Hey guys, so welcome. So last time we were learning about cell multiplication, DNA multiplication, and basically we covered the whole topic regarding that. And so, wow, uh, I thought that maybe I have like one or two views, but it's okay. So today what will we cover? Okay, so today we'll cover these two poems. So let's start with these. How can we fight these? Spontaneous DNA damage. Okay. Okay, so we are going to see how we can fight these and yeah. So we are going to see how can we avoid these mitochondrial DNA mutations. But okay. I think we talked about these replacing missing building blocks, nucleotide supplementation. Nucleotides help us to build the the DNA and RNA. Fixing gene editing. Oh, okay. Mitochondrial DNA mutation of heteroplasmic, meaning a cell has a mix of everything from mitochondria and multiplasmic 41 skills. Fine. Okay, so here we have adding more nucleotides as the first solution. Uh, then we have gene editing. Yeah. So gene editing basically is like using a mitochondrial base editor, and then it goes and finds like a sequence of genes that are faulty, and it changes them to a beta state. So like let's do like DNA transcription video, like making DNA into protein. So I want to use like this video to show you the structure of DNA, and then from there.
The basic unit of all living genes. These genes contain the instructions for making proteins.
So like on a particular DNA, like maybe this sequence is wrong. Maybe here there should be but it should be a T or a C or a G. So basically that one comes and removes the wrong one and puts the right one. So remember we learned about the double helix structure of the DNA. We learned about this that we have the nitrogenous bases, which are these ones in colors, and that they always pair in a specific way, like a C with a G, a T with an A. And basically, when something is wrong here, it comes and patches it. So maybe let's go and see what is mitochondrial base editor. Let's see if you can find a video about it. Okay, gene editing.
What gives people power? Money? Status? Well, they might give some people power, but what gives all of us power is our mitochondria. Referred to as the powerhouse of the cell, the mitochondria is involved in aerobic respiration that generates energy for the cell. It really is a fascinating organelle, and it also contains its own DNA. This is really interesting to understand, but one of the problems associated with this is like the nuclear DNA, mitochondrial DNA can also get mutated, and this can lead to disease. Understanding these diseases and just the mitochondria in general would be greatly aided if we could manipulate the gene so that you could study the mitochondria. But to do this, you would need a tool that would enable you to easily edit the genes. This recent Nature paper may have uncovered a unique way of editing mitochondrial genes. So, hello and welcome to the Shiki Science Show, where in this video we'll talk about this paper and explore how you can actually try and modify genes within the mitochondria. So, firstly, we'll talk about gene editing in general and about the key factors you want in a good gene editing tool. We'll then talk about the development of this tool in terms of the mitochondria, and we'll look at some of its strengths and some of its weaknesses, and then also some of the potential applications of this tool and areas of further study to further optimize the tool and to also discuss where it could go in the future. So let's get to it. If you are interested in understanding gene editing, likelihood is you've heard of the term CRISPR. Usually referring to the CRISPR-Cas9 system, CRISPR is a system that can edit DNA by introducing a double-stranded break and enabling the cell to undergo repair mechanisms, whereby it can result in insertions or deletions into the site, or if introduced with a double-stranded DNA template or a single-strand DNA template that contains desired changes of interest, and this is referred to as precise genome editing. And so either is used depending on the application. And so there are two kind of criteria that are important for developing a genome editing technique. The first criteria is that you want it to be safe, and secondly, you want it to be efficient. Double-strand breaks are notoriously unsafe for a cell, and so there's been much optimization to CRISPR-Cas9 systems to try and improve the safety and also the efficiency to go along with it. And that has resulted most recently in the development of prime editing. But to be able to modify mitochondrial DNA, an alternative strategy was required, and this is because CRISPR-Cas9 systems depend on a guide RNA that targets the CRISPR-Cas9 complex to specific regions of DNA. But getting the RNA into a mitochondria is pretty unfeasible for a cell. Secondly, the mitochondria lacks efficient mechanisms to be able to repair double-strand DNA breaks, and this is another reason why CRISPR would be ineffective for editing mitochondrial DNA. However, CRISPR isn't the only gene editing system. Before CRISPR, there were TALENs and zinc finger nucleases. Similarly to CRISPR-Cas9, these two different editing techniques also induce double-stranded breaks. However, the key difference is that CRISPR-Cas9 directed the complex by a guide RNA. Zinc finger nucleases and TALENs required protein-DNA interactions. So this circumvents the issue of not being able to get RNA into the mitochondria because a protein-DNA interaction can be used instead. However, as I just said, these techniques also induce double-stranded breaks for their gene editing, which, as I've already mentioned, mitochondria don't really have any mechanisms to repair double-stranded breaks. So the question is then, how can you actually edit without double-stranded breaks? Well, the solution to this problem was base editing, which was devised by David Leo's lab, who unsurprisingly are also behind this recent paper.
I think this video is like going to many things. But let's see a summary of it. Okay, this is too much for me to understand. Let's see. But basically, without maybe going very deep, we use these mitochondrial gene editing to fix like a typo. Like if there was a T and we need a C, it goes there and it fixes this. So we are doing all of these to help like the brain cells live longer, uh, like to avoid the radicals, to avoid the fact that there might be imperfections in DNA replications inside of our brain cells that can lead to cancer. And so how do you spell cancer? Let's see. Cancer. Cancer. It's cancer. So transcription errors caused by damage. Damage caused by transcription errors. So remember, transcription is the process of making like RNA. Is transcription the process of making RNA or uh, translation and transcription? So remember, you have DNA transcription and DNA translation. So let's try to see what's the difference between them. Okay. So transcription is making the messenger RNA, and translation is basically taking that messenger RNA and making a protein. So now I think it makes sense. Okay. So to avoid the errors in this stage of transcription, we will have to like open up the DNA. We have to embed toxic PARP1. This is basically it helps to repair a section of damaged DNA, but sometimes it like over it's like an alarm system. It stays on and damages the whole process. We saw this in the previous video. I think we saw this in part 3. Maybe I would have to add more details on everything here. But today I wanted to dive into like the surgery that would be needed. Yeah. So let's say at one stage maybe the only option is like doing surgery and conserving the brain in an external support system, like maybe a robot suit or something. And then this is the surgery that would be involved. So let's go quickly through this full document. I'm waiting for it to load fully, and yeah, we'll go through it. So first of all, before doing anything, we would have to lower down the overall temperature of the brain from like 26 down to 18. This allows the brain to survive on its own sugars. The brain already has how can I say? The mitochondria. The brain has already like a food storage. But if you lower the temperature down to 18 degrees Celsius, it can last from 45 to 60 minutes. And current feasibility of these this is pretty much they're already using it. This is the medical references. And they do this to allow us like 45 to 60 minutes so that we can transfer the brain from a patient to like we disconnect his blood supply and they connect to an external machine based blood supply. So we have enough 45 to 60 minutes. Uh, then we go ahead and dissect the dura mater. And the dura mater, dissect the breast outermost. Yeah, this is currently possible. And basically, dura mater is like a whole mess of brains, of like the tissue to connect the brain dura mater to the skull, to the bone of the skull. So we'd have to be very cautious to like separate those two. And current feasibility is possible because we have the laser tools like high frequency sonic scalpels. Oh, yeah, my bad. It's ultrasonic scalpels. What is ultrasonic scalpels? Let's maybe see a picture of how they look like. Oh, they are just okay. They are just converting electrical energy into rapid mechanical vibrations to cut tissue like precisely. Okay, they are like these things. Okay, makes sense now. Okay, so it's like use big monitor, and then there's like some device attached to it. Like, see here, I think this is like the control unit, and then this is the one that does the job, like that cuts stuff. So so let's summarize this into, let's use a very very simple English to understand this. English to explain the below to a 5-year-old. Use okay. So let's see. Of gel. Oh, okay. To get it, to get to it, you have to open the hard shell. The scanner protects. Right under the hard shell, there is tough stretchy bag that holds the gel all together, just like plastic bag. Okay, yeah, it makes sense. So the duramater is like a plastic bag holding together a wiggly water balloon. The bag is glued to the side of the hard shell in few spots, so you can't just take the gel out. If you try to pull it out, okay. So this bag, our duramater, is like glued on some spots to the hard shell, which is the skull. So you can't just shake the brain out. You have to cut it. Okay. There is the jaws in the okay. This oversimplified things. Uh, yeah, but I think you get the whole idea. Basically, is to be so cautious and we make sure that we remove it. Now, remember, you have 12 cranial nerve pathways. It's like 12, do you see the backbone? We have like 24 big nerves that move from the brain down to the backbone. So we would have to, I don't know, maybe leaving them, it would be advantageous. Um, or maybe we would have to cut them to. But the thing is, each cranial nerve has like a specific function. So if you preserve it, you can read the signals from it and you know the intended action uh, that was supposed to be done by. Maybe I wanted to move my arm. So if I leave the cranial nerve responsible for this movement intact, then it's easier for me. Uh, but like, let's suppose we needed to cut them. I think that would be straightforward. Like, let's say we don't have advanced ways to like preserve them, so we'll have to cut them. But yeah, delete the brain from the skull build to pairs of cranial nerves must be systematically severe because okay, it makes sense. So yeah, because basically these 12 pairs of cranial nerves they move down, then they branch out into many different pieces. It would be a nightmare to like put sensors on all of them, so we'll just have to remove them. But we'd have to be very cautious, otherwise we could damage the internal brainstem nuclei. Okay, it makes sense. Yeah, but if you remove this makes sense. So current feasibility, this is already being done. Like when they want to remove a tumor, they should of course cut it off from the nerve. But cutting all 24 of them simultaneously would lose all the senses. So let's see which senses would be lost. Are they saying that the arms and legs would still work while we cut off the? Oh, cranial nerves, they just go to facials. Oh, really? Um, and the spinal cord is a separate part. Oh, so the cranial nerve and the spinal cord are two different things. I'm sorry, I didn't know that they're just two different things. This is funny. Oh, okay. So the cranial nerves basically just go to the face, ears, nose, mouth, and throat. Oh, okay. Ah, would have to cut a lot of things. So I would make sure that I have to cut these very carefully. Otherwise, if you damage anything, someone can lose, the sense of smell. By the way, why do you need smell while okay? Honestly, at this point, it's so hard. Like, let's suppose we are moving the brain to put it in a robot suit, and now all of a sudden, this thing need smell. Or how can you communicate smell? Maybe for hearing, you need it, and there are like mechanical system that can achieve that. Like they listen, they send like a signal to the brain so that the it understands what's being said. Maybe. Uh, oh my God, this would be a nightmare. Oh, the brain is very very complex. Actually, when I started thinking about it, I never thought about how how this is so hard. Like it's so hard to achieve. But what if you just like maybe preserve the whole head? Because maybe it would be far easier. Or would it? Okay, so yeah, that's it. Circle of Willis cannulation vascular transition. The ultimate of the brain during instruction dependent transfers blood supply. The brain shoots blood through the four primary channels. Okay, so basically this is like, if we ever cut off the blood supply, like when we are moving the brain, you have to immediately connect artificial, uh, blood supply. I inserted that into the artery secured using microvascular bulldog clamps. Okay, yeah, it makes sense. Okay, so let me do something. So, oh yeah, also you have to like make sure that the pressure is uh, average to what the brain is used to. Otherwise, it would be a nightmare. Would burst all microvessels inside of the brain. So the current feasibility is like they have never like done uh, these to four main arteries of the brain like at the same time, but usually they do like one or two at once. But this is still in development in the laboratories. Hmm. Now we are going on section 2. So the five micromolecular repair and maintenance parameters. So yeah, what's this? While the brain is also settled into a biotic change, its cell will slowly decay unless five distinct molecular maintenance tasks are done. Okay, yeah, it makes sense. Basically, the brain cells they need oxygenated blood. They need. Why do we need to change this? Okay, we'll learn about it. So this makes sense. I copied these and now there are some weird things, but I think you get. And this is currently being done, like you can supply the brain with the oxygen. And now let's see microglia depletion stem cell grid replacement. Microglia. Yeah, microglia. Microglia. Let's see how to read this. 1924. Microglia. 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 Ah, mic- microglia. Microglia. Hey. So all of these are around microglia. Microglia. Okay. So, microglia, the resident immune and maintenance cells of the. Oh, okay, makes sense. They are the immune and maintain the central nervous system. They basically protect our central nervous system, and as a cellular cellular damage, okay, tunnel inflammatory and begin to actively destroy the rubber as a periodic paginal cycle that supports cells. Oh, okay. So because over years they start to damage, they hurt these synapses. A synapse is like uh, a place where two neurons like meet. What is a neuron synapse? Yeah, it's like a gap between two neurons. And basically, when they are like firing together, when some billions and billions of those synapses are firing, like exchanging information, that's how you can think, you can reason, you can recall things and everything. So when these microglia are aging, it starts to destroy like your thoughts. Like you start to forget things, you start to not be, your brain is weak. So we need a periodic um, system of like reloading, like introducing new ones. Um, okay, this is how we'll have to remove them. So there is colony stimulating factor 1 receptor that can be introduced to the brain, and then these chemicals strips the survival signals from the old micro, microglia. microglia. So guys, give me a minute. Mm, 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 mm. But I have a question. How are we sure? Oh, okay. So we remove it, and then okay, we add. So in these 7 days, someone will be having no immune system in their brain. Okay, this is crazy. So then later we'd have to inject these young and healthy microglia. Um, okay. And current feasibility, this is still in clinical trials, and in mouse it's succeeding. Yeah, and this reduces, of course, age-related brain inflammation. Okay. Now, overexpression, automated DNA damage. Other neural do not divide. Which demands on the original DNA. Okay. Oh, yeah, this is what we were dealing with in the past document, in the previous document. So, yeah, every metabolic trace on the DNA of the neurons causes stress. And basically, they use like a virus that cannot replicate, which is basically not harmful. And this system delivers the genetic blueprints for CIRT6 and PARP1. Ah, do you remember this guy who helps us to fix things? So that we have to reduce him to avoid the DNA fixing like from. Okay, I think we increase him or reduce him depending on the situation. When he's blocking the DNA repairing, we have to reduce him. When maybe he's short, he's there are a few of these, then we would have to increase them. Okay. So basically, if you will learn this, it makes sense that if you increase it and it makes the process of fixing the DNA, a lot um, faster and enoughly. And in mice, they are shown, a significant improve in the DNA repairing processes. Now, what is this? Epigenetic reprogramming. Even when DNA remains physically uncompromised, noise over time, the chemical types of DNA is common. Okay, so this what those proteins to the brain tissue via the cells. This is the first the neurons go back to their cellular age software to ecosystem. This is the division of aging mice. Oh, okay. So you're telling me that even when the DNA is not damaged, due to some the chemical tags on DNA gets scrambled. Hmm. Okay. Explain deeply. Oh, so because like the DNA is so huge and contain information for many things, so it uses chemical tags to know this information is for these, this part encodes for these, you know. Mm. And those are like the chemical tags. They are like bookmarks. Okay, yeah. Okay, the solution to the scientific names. So, so specific proteins for act like a magic for any cell. Okay. Okay. Oh yeah. It makes sense. If we use these guys, they will reset the whole brain. And now you are like born again. Like you don't remember anything. You don't know nothing. So you just do like short, um, give it small, like frequent small shots, like, and then eventually we wipe out the damaged. How can I say the damaged bookmarks, the chemical tags, and the cell makes new ones. So yeah. So let's ask. So periodic shots of small quantity. Yamanaka, is it Yamanaka? Factors is the worn out chemical tags and the cell creates new chemical tags itself. Okay, yeah. So when the cell realizes that it has no okay, yeah. So when we remove the old messy chemical tag, the cell still leads to on DNA, it knows what to do and what it should be. Yeah, so it makes sense. So we'll have to do this like occasionally in small doses to just remove the old damaged links from the brain and let the cells fix themselves up again. So synaptic structure stabilization. Oh, okay. So these chemicals are like fertilizers. They tell the synapses like it's like fertilizing the synapses, like keeping them strong and healthy. Okay, yeah, makes sense. Claiming, aiming. Oh, okay. So this is still in human clinical trials. Hey guys, do you know ALS? So bad, so bad. Like you can't control any part of your like you can't like from the neck down you are paralyzed. You can't do nothing, especially for movements. Okay, yeah, for movements, not for like digestion. Like digestion is fine, everything is fine, but you can't move your legs or arms. This is what they call ALS. And basically in ALS, your neurons vanish. The neurons that should get information from your brain to the hand to like move it sideways, they completely um die die off. And yeah, so I don't understand how this can cure ALS though, because in ALS, okay, so let's ask the below cure ALS, while ALS means is a state where a patient has lost the neurons that were supposed to carry the motor muscle signals. Mm-mm. okay, so in ALS we lose the connection because they were never being used in the first place. Ah, it's like if you are not, if you were a dancer and you get into prison and you no longer dance and you lose the ability to dance, like you forget how to dance because they need the connections that were supposed to keep you um able like know how to dance. They are not in use, so they just uh, waste away. Okay, they like die like simply, and because they're not being used. So now how does BDNF and NT3 cure this? And neurotrophic factors, think of them as benefited nerves. Oh, so they keep these things active so that they don't wear off. Okay, makes sense. Still there, they make the meningetic neurons work harder. Oh, okay. So BDNF basically tries to help the disease not to like spread like wildfire. It slows it down. Like in ALS, you lose control like over time. Like maybe in the first month you can't do like 100 push-ups, but you can do 50. The second month you can only do 10 and feel very tired. In the next one you can only do like one. Then after like a year you can't even move your arms. So basically, if you are taking this BDNF, it slows down the process that can, you can continue being physically active, but it's declining, but at a very slow rate compared to not taking the BDNF, because the BDNF essentially forces the brain to keep those connections, the forces the neurons to keep the connections, and then you lose auto activity when those connections are not there. So BDNF is like connection to keep alive, don't give up. So yeah, now we understand what we have to do. We'll be using this BDNF to make sure that all connections in our brain stay intact and functioning, to not lose any connection or functionality at any stage of our life. Section 3, the biohybrid and the manufacturing plant. Building Oh, okay. So basically, the idea is that um, building a whole system that will create the liver like it would be so complex. So maybe we would have a robot suit, but that robot suit has like a liver somewhere, kidney, another place. You get what I'm saying? Okay. Yeah, and also have to do this in that organ that we are gonna use as a donor organ. We have to remove alpha gal, which are surface sugar molecule that triggers the human immune attacks. Okay. And instead, we put the friendly human complement regulatory genes. The living organ block function as the ultimate natural filter clears out. Oh, okay. It's like having a liver and then a kidney alongside the robot chassis. Oh, so there are people living with pig kidneys and hearts. What? Is there a person who lived with a pig kidney? Oh, what? Oh, this is interesting. Damn, I didn't know about this.
We sat down with Tawana exclusively as she shares her hope for others.
I could feel the blood flow through my veins. That's how strong the kidney was pumping.
Tawana Looney is only the 5th person in the world to receive a genetically modified organ from a pig. She's part of a growing group of trailblazers undergoing groundbreaking procedures, which doctors say could help change the world.
It is, I think, a very important component of a multifaceted approach to trying to relieve the organ shortage.
Recipients right now are approved on a case-by-case basis.
If you were to put an unmodified farm pig organ, whether it's a kidney or heart, into us, we would reject it in a matter of minutes. So we're actually adding a gene that inhibits the immune system right to the organ.
The first 4 recipients all passed away within 3 months of their transplants, but Looney is so far the healthiest person to receive a pig organ. She spent more than 7 years on dialysis and 5 years waiting for a human kidney. But her blood was so full of antibodies, she said her body would have rejected hundreds of potential matches.
In 2017, I heard on the news of this research. Oh, it just gave me so much hope. You know, it just did. I said, I want to do this, I want to try it.
This isn't the first time Tawana has been involved in a kidney transplant. Back in 1999, she donated one of her kidneys to her mother, and now she hopes her latest procedure will help future generations.
It's just like getting a second chance in life.
And doctors say Looney is still being monitored, but is doing incredibly well. The next big step is for the FDA to approve human clinical trials, and if that goes well, these procedures could expand on a wider scale. But she looked great, she was doing great. I mean, I honestly felt like she could do laps during that interview. Wow. I mean, she had so much energy.
Because you've been covering the story for some time.
Yeah, we went to the research farm. We also saw the research lab exclusively as well. And it was really incredible to see scientists are really on the verge of a breakthrough here.
Very encouraging. All right, thank you, Morgan.
It is now with great pleasure that I introduce our second student speaker, Lois Olabi. Lois was born in southwest Nigeria and raised in southwest Texas, and she appreciates fufu as much as a good breakfast taco. She is the firstborn daughter of Nigerian immigrants, who I'm looking at today, such a beautiful family, who raised her on a community education and service, values that carried her into medicine and that she carries now into every room she enters. Lois and I spent our clinical year together in a small cohort, so I can tell you what the CV does not. Lois is brilliant, but what makes her unforgettable is the way she makes people feel protected. Patients
Oh, I was muted this whole time. This is funny. But basically, what I was saying is, okay. So yeah, okay. So basically, we saw that there is this woman who has been living with a pig kidney, but eventually she developed like an infection, and they had to remove it. But it shows that it's possible. And also, yeah, I would have to make the white blood cell, like the macrophage and T cells. They're like the army that fights off diseases in your body. So we'd have to make something that continues to make these guys and pump them to the blood. So hematopoietic blood producing structures. Oh, so these hemato. Okay, I'm giving up. I'm giving up. Hematopoietic. Oh, what? We don't have this word in the dictionary pronunciation. Pronounce. Hematopoietic. 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 Why is this Y? Hematopoietic. AI overview. Okay. So basically, we will need this continually make this macrophages and T cells. But I wonder, the ones that we already have, are they dying off? What do they mean by primate models, though? What do they mean by primate models? Why do we need to replace T cells? Are old ones dying every day? Oh, so they continue to die in a war. Oh, eh. Okay, so the more I learn facts about our body, like the more I respect it. Like, oh my God, the body is so brilliant that it knows that some things are not meant to last long. Like these cells, they are like soldiers. They carry active weapons. So if we don't stop them in time, they might even end up. Okay, I just got distracted. I'm sorry. Okay, so this soldier, they die within like 7 days. So yeah, we need to replace them. Okay. Yeah, they have to be human compatible, of course. So now this makes sense, and it's being. Oh, so what do they mean by? Oh, okay, so this is good. So they took genetic pig bone marrow, they put it to monkeys and successfully grown human compatible T cells in those monkeys, and those T cells controlled and patrolled the the blood and brains of the monkeys, and it was successful. This is amazing. Oh my God! Imagine taking a pig bone marrow, putting it to monkeys, and producing something to help humans. Like this is crazy. But like, is it good to continue using other animals to benefit ourselves? Using pigs and monkeys to build things to help us? Is it like ethically or is it religiously accepted, or is it ethically or morally right? I don't know. You tell me. Okay. So let's resume section 4 next time. I have a few things to do today. So yeah, see you, and it was fun learning all these things today.